This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 238. Recorded on March 18, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
Is it nice there in San Diego?
Actually, it's a nice day, yeah.
Yesterday, we recorded a TWiV with Forrest Rower.
Oh, very good.
You know him, right?
It's San Diego State.
He said it was a little chilly there.
Yeah, it was a little chilly yesterday. And you had Maria Rojas.
Maria Rojas, very good.
She's from Ecuador.
Yeah.
My old country.
That's right, your old place. That's right.
Yeah,
she was great.
She's a great kid.
They did a very cool citizen science sampling study where they sent people swabs and they had them swab and then they looked for SARS-CoV-2 RNA. Very cool. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. By the time this episode posts, it will be spring.
21st of March, that's right. Are you happy about that?
I sure am.
Good. And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Hello, Michael Schmidt. Is everything just wonderful in Charleston?
No, we have a line of tornadoes coming through as we speak. We're getting that fall weather that's devastated the Midwest over the last few days with horrific tornadoes. And we canceled classes at noon today and sent everyone home because they didn't want people to get stuck in the flood.
Wow, that's pretty serious. You think it's going to materialize?
It's raining right now and it's dark. So
well, I hope it's okay. Michael, these classes are in persons already?
Some classes are in person. Our esteemed governor has declared COVID over, and we can go back to, everybody in restaurants, and it's unfortunate that we're supposed to maintain our social distance and do everything like we're supposed to, but he wants the state open.
Yeah. Okay. I guess he has he or she. I don't know.
It's a he, Governor McMaster.
The governor has a degree in epidemiology, I understand. Is that correct?
No.
Oh, okay.
No, the governor is a lawyer, but he has been relying heavily on the state. Our state public health and control. And so we never had a mandatory mask order for the state. It was left up to the local towns and cities to effectively do masking. And, for the most part, the towns and cities have been following the masking guidelines. And
good.
We're hoping that the public and the restaurants and bars continue to do their part, because for the first time, we did see a drop in the number of cases.
Very good. All right. Did everyone enjoy last episode of TWiM? Our little,
that was great.
We got some nice comments. And the one thing I forgot to do last time was to thank Ray Ortega for work. He's been here since the beginning of TWiM, and he's helped to edit and post every episode. So Ray Ortega works for the American Society for Microbiology, and he is out in California. And we thank you, Ray, for all your work with us.
Thank you, Ray.
Thank you,
Ray. Actually, so Elio, Maria Rojas reminded me that she was present when we recorded at TWiM at San Diego State, and we talked all about your career.
Yeah,
and Michael and Michele were there, and she was in the audience. So I dug up a photo, and there she was. She said it was so nice to watch it being recorded. Really nice. Ecuador. Okay, so today we're back to business as usual, and we will have first from Elio is going to tell us about a recent virtual visit to Israel.
Yeah. Well, here's the story. I was asked by a professor at the Hebrew University in Jerusalem, an agricultural microbiologist, essentially, Amir Levy, and he asked me to give a journal club on Zoom. So I thought about what can I do, and I came up with something very different. I came up with, he asked me, would I philosophize a little bit? Would I talk about where microbiology stands? So I said, okay, I'll do that. But then I would just as soon tell stories from the blog we have, Small Things Considered, which has some pretty amazing tales. In fact, some of the tales are not even in the blog, but that's okay because they're amazing stories. So that's what I did. So if you want to hear what it is I said, I can tell you.
Yeah, love to.
Okay.
The images are amazing.
So first of all, the first part was to discuss what are the big themes of modern microbiology. I call them big themes instead of saying what is the most important thing in microbiology today. I don't know that. That's too complicated. But themes are just saying something you choose. So I chose 3 themes, and the themes are the following. Number 1 is metagenomics and other metas like metatranscriptomics has totally revolutionized our understanding of ecology. In the past, if I gave you a sample of feces, soil, water from the ocean, and said, "What's there?" you had to culture it, streak it out on a agar plate, and see what grew. And that's how you could tell what's there. And the problem is that even though the bacteria associated with the human being, about half of them can be cultivated. Out in nature, the number is about 1%.
Mm.
So by doing cultivation, you're missing completely on what's there. But if you look at what's in the DNA, you can more or less tell what's there because that's easy. That's what metagenomics does. And if you want to know what are they doing, you can do metatranscriptomics. So this is a complete revolutionary way of thinking and of looking at. So it's a big theme because everybody's doing this. This is now the accepted way of studying microbiology in a natural sample. That's number 1. Number 2, and they're not really related subjects, is the advance in microscopic techniques. And there are 2 classes, or maybe there are more, but they can divide them up into cryo-electron microscopy, which means you can do a section like a CAT scan, that is a section through a bacterium, through a cell, through anything, and you can then reconstruct it in 3Ds. So you can tell, and this is done by freezing the sample so rapidly that nothing can happen. So you're immobilizing the structure of let's say, a bacterium, and you can cut through it if you wish and look at it, and that's amazing. And along with it, it's kind of in parallel, is the development of fluorescent techniques. And then fluorescent techniques means that if you stick a fluorochrome onto a protein molecule, you can see where the protein molecule is. So you can tell not only what it looks like, but what's there. And that is pretty fantastic. And the third one is a more general thing, which is that we realize in the last, 20, 30 years, that bacteria are social beings. They are not the way, when I was young, which is a long time ago, the thing that one thought is bacteria are all single things. They all swim around by themselves, unperturbed and undisturbed by everybody else that's there. They do not have a social life. Well, that turned out to be very, very wrong. And the social life can be seen in a variety of ways. And what I mentioned was examples. The main one is quorum sensing, which, as many of the listeners know, means that bacteria can tell how many of themselves there are. Well, is there a quorum? And the reason why it's important is because imagine a single bacterium, a single typhoid bacillus or diphtheria bacillus infecting a human being. A single one is capable of making toxins. But the amount of toxin that a single bacterium will make is irrelevant. It doesn't do any damage. So why bother to make the toxin? Making toxin is energetically expensive. So if you don't have to make it, you don't make it. So instead, what you do is you figure out how many of us are there. It's called quorum sensing. Okay, that's one. The other is making biofilms. We know that most bacteria in nature do not swim freely just by themselves. Most of them make biofilms. They make biofilms in the oceans because there are surfaces like seaweed, particles. They cling to that and they make a biofilm. But a biofilm is like a little city of bacterial cells, and that is an intense socializing thing. A lot goes on. Swarming, that is the movement on surfaces by a collection of bacteria, is a social activity. Making toxin antitoxins, that is, bacteria make compounds which affect other bacteria. They can kill them, in fact. And this is delivered often by a thing called type 6 secretion. There's all kinds of types of secretion whereby bacteria can secrete protein molecules, effectors they're called, which can affect either animal cells or other bacteria. So these are examples of socializing. So this is pretty much what I had to say to these people. The audience, by the way, was a bunch of graduate students listening to a journal club. So I figured they should know all this. Okay. So what do you think? You agree with my presentation of themes?
Yeah, I think it's great.
The one
Yeah.
theme that I think that you needed to reintroduce to them is the importance of understanding the physiology of the community. You sort of have it there with omics by metabolomics, but understanding the interaction of the physiology is, I think, the one thing that is missing from many of our graduate programs. We're doing a lot to teach omics, but there are very few folks who have been trained in recent years that understand the significance of the metabolic pathways that were worked out when you were young.
That's true. Yeah, you're right.
And now it's up to this generation to put the metabolic pathways to work to understand how these communities work. And Jo Handelsman sort of dropped the gauntlet or challenge when she talked with us on our last episode. And it's simply microbial ecology. It's understanding how all of this gets together. And we know that today as the topic of microbiome.
Right. And if I could expand on your point, it's not just the physiology and metabolism of an individual bacterial cell or an individual pathogen, but rather the food chains that occur and are developed within a community that stabilize that community.
It's the food web.
Right.
And then the other thing that any self-respecting microbiologist needs to know today is they have to understand the host side. So they need a good foundation in immunology.
It's called ecology.
Yeah, that's it, Elio.
Understand the environment in which you are.
Yeah, that's it.
Elio, did you do this in English, I presume?
I did it in English, and I had to explain the following. I started out by saying, "Ani lo medaber ivrit," which means I do not speak Hebrew. But I explained why. The reason I don't speak Hebrew is that my mother was a Hebrew teacher. And if your mother is a teacher of a language, you're not going to learn that language. There's no way. So I never learned Hebrew. But they all speak English. I mean, so it's not a
Did
problem.
they enjoy what you said?
They say they did. we talked a lot. We talked a lot about things which were not quite relevant to what I talked about. Namely, they want to know the graduate students, when beginning graduate students, how do you choose a thesis topic? Okay. Well, I have one little thing to say about them, and that's the following: if you're going to go into research, and I don't know how many of these people in the audience intend to go into research, but some do. Let's say you want to go into research, or you finish your PhD, you do a postdoc. By the time you've done that, 8-10 years have passed. What at that point, from the beginning, when you're starting out, how do you know what's going to be hot in 10 years? You have no clue. So it doesn't matter. To a large extent, it doesn't matter what the subject of your thesis is. What you want to do is to learn how to think, how to deal with it, and above all, be flexible. Learn how to adapt. Learn how to choose new and wondrous things that are coming down the pike. So don't agonize so much about what the subject is. Agonize more about what kind of a lab it is.
Yeah.
Is it a lab where you are a peon and just do what the boss says?
Pair of hands.
A pair of hands. Or are you a place where your PI is actually engaged in what you are doing and how you're doing it? And that's more important than what you're doing.
That's great advice.
Yeah, I agree. You need to find a good mentor, basically. Yeah.
The kibitz factor.
We can't predict 10 years from now what the new and exciting questions are, but we also can't predict what methods will now be available to ask those questions. So,
for sure. So, Elio, if you were doing lab work today, would you be using some of these approaches?
Oh, sure. I couldn't help it. They'd be forced on me.
It'd be forced on you, yeah. Well, but you would probably still do, in virology, there are old established techniques that we do still, and many new people in the field don't use them, right?
Plaque assay.
But plaque assay is one of them. I think it's a very powerful.
Of course. Modern doesn't mean that the old is gone.
Yeah. It's, I think they're very informative, but few labs train people to do plaque assays. They do PCR, which is not the same thing. It's totally not the same thing. So I guess it's the same in microbiology, right?
Sure.
I have a question. If we were to add a fourth theme, do you think it's that science is more interdisciplinary, in part because of the specialized techniques that we can now apply?
Well, yeah, but today's interdisciplinary is yesterday's single subject. We take it for granted. Obviously, using different kinds of microscopy, using different kinds of, especially the kind of mathematics which you have to use. All of that is very interdisciplinary. You cannot be a biologist today without being a biomathematician, for instance. So, yes, the answer is of course.
All right, Elio, thank you for that. Appreciate it. Now let us move on to a manuscript, and Michael's going to tell us all about that.
I'm going to try. So the title of today's snippet is Meta-Analysis of the Parkinson's Disease Gut Microbiome Suggests Alterations Linked to Intestinal Inflammation. And this was from Romano, Sava, Beddarf, Charles, Hildebrand, and Narbad. And they're from a number of institutions spanning from the Norwich Research Institute, the Department of Neurology in Bonn, Germany, at the University of Bonn, the University of East Anglia, as well as the Norwalk Research Park. And, well, as the title implies, this paper has done a deep dive for us. Asking
what? Say it again.
A deep dive for us.
Okay.
Taking us to this fundamental question of whether or not there are remarkable differences in the gut microbiome of individuals with Parkinson's disease versus similarly age-matched controls. First, since this is a show about microbes, I should at least define Parkinson's for our listeners. I must admit that I did not appreciate the significance of this disease until I began to get into this paper. And first, Parkinson's disease is the second most common neurodegenerative disorder after Alzheimer's. Globally, it's got an incidence rate of 10 to 50 new infections per 100,000 population per year, and a prevalence rate of between 100 and 300 individuals per 100,000 people. The scary thing is this number is anticipated to double by the year 2030. So this is big news, so to speak, because there are going to be a lot of people with this ailment. The pathophysiology is such that Parkinson's disease affects predominantly dopamine-releasing neurons of our brains. And that's implicated in the disease, leading to decreased dopamine levels and motor impairments that I can appreciate because I had an uncle with Parkinson's, because he had a pronounced tremor. There was some rigidity in how he walked, and he had balance difficulties. And he had a challenge with spontaneous movements. From a molecular perspective, there is an intracellular deposition of the aggregated alpha-synuclein. Now, I had never heard of this protein, but it's a protein of unknown function found primarily in neurotissue, making up as much as 1% of all the protein in the cytosol of brain cells. And unfortunately, in Parkinson's, what it does is it misfolds in aggregates into clumps that the pathologist refers to as Lewy bodies. It's thought that these Lewy bodies may be toxic and remarkable in that these aggregates may get shunted from neuron to its neighboring neuron, causing the spread of disease.
It sounds like prions. It sounds
yes,
like prions.
It really does sound like prions. So, which leads the brain to enter a neuronal cell death and neuroinflammation, which long-term listeners of this podcast will appreciate. Anytime we say inflammation, or anytime inflammation enters into our mix, you can appreciate that dysbiosis or a difference in the microbiome is somewhere in the mix. And finally, Parkinson's disease is often considered a multisystemic disease affecting the central as well as peripheral nervous system, resulting in several non-motor symptoms, often including gastroparesis, which is, in layman's term, constipation. And the GI symptoms often are a tell. They often can precede movement disorders by years. Additionally, there are deficits or alterations to the enteric nervous system. This is the thing that controls our gut, which is comprised of about 500 million neurons of our autonomic nervous system, where this mesh-like network, I view the enteric nervous system like the World Wide Web, governs the GI system independent of our sympathetic and parasympathetic nervous system. And to really put how important the GI nervous system is, it's got 5 times as many neurons as our spinal cord.
Wow.
And here's the joke of the day. And it's got about 2/3 of the neurons found in Alan Dove's cat.
What?
Alan Dove is a co-host of TWiM,
Oh.
and his cat is continuously trying to ham it up in the background. And finally, these neurons are derived from the neural crest, which is the same genesis organ that gives rise to our brain. So with the flurry of activity of everyone delving into the microbiome surrounding chronic disease, it should not be a surprise that investigators have looked into the obvious question for differences between Parkinson's and normal controls. And it's indeed been investigated by 22 studies, And Michele alerted me to the fact that I remember back in 2016 reading a paper that came out of Rob Knight's group that appeared in Cell. And I'll toss that into the show notes for people because it really goes to the point of why there have been 22, if you will, clinical trials looking at the microbiome of Parkinson's to controls. So in this papers, the authors conducted a meta-analysis, which again, if you go back, you effectively look back at previous studies and you assemble them and ask the question, what's common? And when they went off and looked at these 22 studies that were done, only 10 of them had made available the data that they used in their studies. And the data that they used were 16S ribosomal RNA gene sequences. And they specifically looked at the microbiome from Parkinson's disease patients and age-matched healthy controls. And in total, they looked at about 1,200 individuals. So this is a 1,200 individual look back from these 10 studies. As you can likely surmise, anytime you have 10 clinical trials, they're not all done the same way. We've witnessed this with the SARS-CoV-2 vaccine trials. Not everyone was done the same way. And this was true for these 10 separate studies of Parkinson's. 9 of them were effectively unique, but you're going to say there's 10. Well, one of the studies looked at an initial screen, and then they snapshotted it 2 years later in a separate study. So there's a lot of work that preceded this meta-analysis. Bottom lining the microbiology, they found significant microbiome alterations in folks suffering from Parkinson's disease. The interesting thing is that the meta-analysis offered these authors an opportunity to tease out study-specific irregularities, heterogeneities, whatever word you want to use, that can help impact our understanding and our ability to compare and contrast, as they stressed that though the differences in the microbiome structures between Parkinson's communities and controls were significant, they were small. You know, they're trying to make sense out of what significant really means.
Looking for those bright, shiny
how
needles
consistent were they
in a haystack
between populations?
Pretty well. And we're going to get to that in a minute. Principally, microbiologically, in Parkinson's disease patients, and that was the beauty of their meta-analysis. They were effectively asking Elio's question, how close were they? And that's really the beauty of this meta-analysis, and candidly, why I picked this paper to discuss today. I think now that there have been a number of microbiome studies out there on everything from asthma to Parkinson's to you pick your favorite chronic disease that folks are looking at, I think we need to do a look back and begin to do what this group did to give us some insight into this disease process. So microbiologically, in Parkinson's disease patients, they saw an enrichment for the following genera: Lactobacillus, a gram-positive rod known for its ability to drop the pH of a local environment to around 5. Any student of microbiology knows that a pH of 5 is not very friendly to the garden variety gram-negatives running around in our gut. We like neutral pHs. Secondly, Akkermansia, a gram-negative strict anaerobe that is capable of consuming mucin, that snotty substance that serves as a protective layer around our intestinal wall. it serves as a source of carbon and energy for Akkermansia. And that in turn, its waste product of Akkermansia is short-chain fatty acids, which in turn feed the enterocyte, the epithelial cell that's lining our intestinal wall. So the waste product actually maintains our intestinal wall. So Akkermansia was enriched, and in point of fact, Akkermansia can account for 4% of the intestinal bacteria in healthy controls. And Akkermansia was enriched in the Parkinson's disease patient. The other caveat, and most folks don't actually appreciate this, and this goes back to another sidebar conversation that Vincent and his friends have been having on TWiM, is that there are many strains of Akkermansia, and strains mean they behave differently. This is not a variant. This is a different player. But the problem with 16S ribosomal RNA sequencing is the ribosomal RNA is likely the same. So you're not going to be able to tell the difference between the different strain variants of Akkermansia. And that's what this study couldn't figure out. And again, Akkermansia is no stranger to TWiM. It's been investigated for its association with obesity and diabetes and inflammation. The other microbe that was upregulated in the Parkinson's disease population through this meta-analysis was Bifidobacterium. And as its name implies, it's bifurcated. It looks like a Y. And this is a gram-positive, non-modal, often, again, branched anaerobe. It's likely the first gift you received from your mother as you went down the birth canal, because the hallmark of Bifidobacter is it helps the newly born infant to digest human breast milk. What microbes were absent in the Parkinson's disease population? Again, contrasting them against age-matched controls. Well, they saw the depletion of bacteria belonging to the Lactospiraceae family, who are a family of Clostridial in the order Clostridiales that ferment diverse plant polysaccharides. The reason this is important is the Clostridiales waste product from the fermentation of the plant are again short-chain fatty acids that feed the enterocyte. They principally feed butyrate, acetates, alcohols, and the other organism that was depleted or decreased in concentration in the Parkinson's family was Faecalibacterium, which is a gram-positive mesophilic rod that's a strict anaerobe. And again, it's purported to be one of the most important commensals of the human gut. And again, the deleted microbes are important because of the waste products that are going to be absent. And it's all about this, as Elio pointed out to the students in Israel, it's all about this dynamism that's going on between the microbes. We're no longer having single cells. So what these authors have done for not only the Parkinson's community, but as the microbiology community, is they told us that we have to pay attention to these relationships. And when you read the last line of their abstract, and this is where my microbiological bias comes in, it suggests to me that dysbiosis is really a complicated process, especially when it comes to these complex disease etiologies where there's a long lag before you see symptoms. We initially see the foundation symptoms as constipation in the gut, in a cell that is derived from the same neural crest as the brain cells. So we have to look at model systems. And unfortunately, I had forgotten about Rob's paper until Michele reminded me of it. And I got to go back and reread that. But I think this aggregated alpha-synuclein protein, again, of unknown function, it really gives us pause to, again, begin to think how we need to refine our hypotheses and how we need to begin to design those animal studies in order to get a more mechanistic as opposed to descriptive approach of how the microbiome is influencing a disease process like Parkinson's, which seemingly is simple. It looks like a prion disease where you're getting aggregation. So now let's look at how this paper arrived at its conclusions. I believe this is, again, where we're going to see more and more papers like this in the future. So if you're involved in such studies now, make certain your study protocol allows or requires that you deposit your sequence into a publicly accessible data repository. And secondly, you bring along the relevant de-identified patient information. Like, for example, were they on antibiotics? How long ago was their last antibiotic dose? We learned from this paper that wasn't controlled for in some of the studies. It was in some, but not in others. This again gets back to Elio's point of how you can compare and contrast the data. And again, this is so important when you begin to try to look at clinical trials, because not every clinical trial is going to be equivalent in design or patient number or anything else. But these meta-analyses can help us tease out the complex interactions between the host immune system, and the microbiome. And that is precisely what these authors walk us through. They offer, go ahead, Elio.
I've forgotten at the beginning you told us how many different studies did they look at?
I was just going to tell you again. They started by 22, and then they could only find sequence data for 9 of them. And then they actually had 10 studies, but one study and the second study were 2 years apart, but they followed the same protocol. So they just call that effectively a single study.
What was interesting is those 9 different studies were looking at different patient populations from different regions of the world, places where the diet was different. So there were a lot of variables, and they used these sophisticated statistical methods to try to find the shiny needle in the haystack.
And the first table of their data is effectively, they grab the bibliography and they glean, you not only have the reference, but you have how they isolated the nucleic acid, the protocol they use, how they then sequenced it. And all of these things we learn in their study can actually impact on the data that come out.
I found that a little bit shocking.
I was disturbed by that,
Yeah.
which is why we need more papers like this to go back in time and try to recapture the flag, so we don't have to redo the early microbiome data that, gave us a number of what's been going on. The first figure that they give us, you see how many control and Parkinson's patients are in each study. And the Hills and Burns study, which is this third study, had the largest number of patients, 325, which is, a big trial in terms of to being able to recruit and get patients to produce stool samples.
Let me ask you an obvious question. Some of this suggests that fecal transplants may be very difficult to carry out, but still, it's possible that maybe one should try. What's the story on that?
Well, we know that fecal transplants are very successful for eliminating a chronic infection like C. diff, Clostridiales difficile. But we don't know whether or not fecal transplant can reverse a Parkinson's disease syndrome once the pathology has started.
Right.
Which is why I went through the exercise
I you say that.
Well, it's why I went through the exercise of describing the pathology where the Lewy body becomes the agent of change for the neighboring neuron.
Well, you're going to, wait a minute, that's going too fast. Right now, you deal with the fact that the
microbiome's changing in the gut.
And you can try to change it back.
But
wherever that leads you.
But when do you look? When do you look to see if do you base it on genetics? If we believe that your microbiome is genetic and not environmental or diet driven. So it gets us into a number of hypothetical and hypothesis generating types of studies. The second bit of their first figure is their bioinformatic workflow, the secret sauce, so to speak. Those of you new to this game, this and the methods section of this paper bears a look. And you need ready access to a bioinformatic cheat sheet. So if any of you out there have a bioinformatic cheat sheet, send it to TWiM. I could have used it for this paper. As your understanding of this will offer you the insight in how they arrived at these microbiological conclusions, I have already bottom line for you. Bioinformatically, they principally use three methods: the total sum scaling methodology, the variance stabilizing transformation, and the center logged ratios. Fundamentally, you have to understand that this is all in the realm of non-parametric statistics because everybody's different. You can't do an analysis of variance because of the complexity of what's involved. And the bioinformatic approach typically that I've been familiar with is looking at alpha diversity, which is really the average numbers of species in a particular habitat. Here in this case, the gut of a Parkinson's patient versus the gut of a healthy age match control. So alpha diversity, if you will, is a local measure. Beta diversity refers to the ratio between the local or this alpha diversity and the regional diversity. And this diversity of species between two habitats and regions. Compare the diseased versus the control. So again, what I found most interesting is how they were able to make their inferences. And their data suggest that initially that the gut microbiome between the diseased patient population and the control is different, and it's significantly different. Again, as I'm not as deft as I would like at describing the stats that they used using audio alone,
Yeah.
I'll defer.
Please don't.
I'll defer to what the authors offered from their discussion, was principally that their analyses suggest that the particular methodological approaches, such as sample collection, how the sample was transported to the lab, whether it was frozen, the sequencing platform, and then, curiously enough, the chosen region of the 16S ribosomal RNA gene that they looked at. Remember, the ribosomal RNA gene has V1 to V2, V2 to V3, and that could account for some of the reasons for the heterogeneity between the 10 studies. And while each of the studies had some differences between their bioinformatic approaches, they believe that the most robust and comparable results were obtained when using the V3 for V4 region, with, if you know anything about anaerobes, is the preferred 16S ribosomal RNA target for seeking anaerobes. And they take us to the confounding factors. And again, they point out that the Parkinson's subjects were likely on medication. And levodopa is what most of the patients were on, and they don't know whether or not that could have influenced the microbiome to deplete the microbes that are there, because the ones that were the enriched and the ones that were reduced are all considered good guys from a probiotic perspective. And so we don't know what's actually going on. But that's not to say all is lost from their meta-analysis. By stratifying the analysis by study, they could simultaneously account for variability introduced by the study, intra-study specific factors and methodologies, as well as country of origin, DNA extraction methods, and which takes me to their last figure, figure 6, which gets back to how we started our journey on TWiM today, thinking about microbial ecology. And they looked at the metabolic pathways, and it showed a significant difference in abundance of the metabolic pathways contained within the Parkinson's disease microbiome and the control population. A piece of advice when you look at figure 6 is blow it up to the size of a house, because you can see the fine differences between the sliding bars. And it will help you decipher who's up and who's down, and then tap the shoulder of your favorite microbial ecologist and physiologist to help you understand the relevance of what's happening and what's significant, which was, again, this is all based on hypothetical functional predictions based on 16S telling you the name of the microbe. And so what they did is they said that based on the population, the Parkinson's disease microbes were enriched for pathways that were related to ubiquinone, or coenzyme Q, menaquinone, which is vitamin K, biosynthesis, glutamate degradation, methanogenesis, and lactic type fermentations, which makes sense since lactic acid bacteria were enriched. The controls, on the other hand, were enriched for pathways involved in vitamin B12 synthesis, and again, glutamine rather than glutamine and glutamate degradation, and the degradation of glucuronidate and galactoglucuronidate and methane production, not via hydrogen, but via acetate. So what the microbiology is telling us is that this enrichment for Akkermansia, and the other studies have found that Akkermansia is often enriched in other neurodegenerative diseases, is that it may be tied to the degradation of mucin. The sentinel event. As you increase Akkermansia, you may be affecting gut inflammation, you may be altering permeability, which then can go into a higher rate of endotoxin leaking in to circulation, which then will result in systemic inflammation. They offer a lot of on speculation about the role of Akkermansia, but I'll leave that for another discussion. So summing up, their meta-analysis is like a systematic review of, routine human clinical trials. And I'll think it will greatly help us go from descriptive to mechanistic understanding of the role of microbiomes in the development of chronic disease.
Michael, not clear to me if they think that the dysbiosis is a cause or a consequence or both. What are they thinking here?
They're very careful to say that they cannot establish cause and effect. That
Yes.
Additional studies will be required. But if you couple it with this earlier paper, the 2016 paper from Rob Knight and Sarkis Mazmanian, where they take germ-free mice and then they install in the mice fecal material from either controls or patients who have Parkinson's. But at the early stage, they're not yet symptomatic, they're not on drugs. And then they can measure motor function in those two populations of mice, and they actually see more motor defects if the microbiome is from a patient that had Parkinson's. So that is consistent. A mouse, right. So that is consistent with there being a cause. But the authors, to their credit, are very careful.
They're very, very, very careful.
And Michael, it's true that many of these studies are descriptive, but I would also say they are predictive, which is a bit more, has a better connotation. So they're making some predictions about impact of vitamin K2 or vitamin B12 or whatever. And then we can go to mouse models or other experiments to test those ideas directly.
And that's I think the value of papers like these is that they're going to go from these informative to enabling us to postulate a mechanism, then design the clinical trial that can test whether or not the hypothesis from the animal study warrants a full-scale human trial.
All right.
Let's hope so, because if Parkinson's disease prevalence is going to increase as the elderly population increases, we need other methods to manage it, and it would be awesome if we can do it through diet and perhaps fecal transplants.
And I had no idea that acromantia was implicated in things like MS as well.
Right. Another inflammatory
disease process.
Mm. All
right. Thank you, Michael. Let me read a few emails before we wrap up. The first was from Stan Malloy. Every episode of TWiM is great, and I really enjoyed your 10 years show as well. Thanks so much for everything the TWiM team does to make the show fun and exciting, and particularly your insights to bring the TWI series to the world. Stanley.
That's great.
Thank you, Stanley. Holly writes, good morning. My name is Holly. I'm a microbiology student at Skyline College in California. I listened to your podcast on microbiology papers for first-year students this morning and really enjoyed it. I'm planning a career in nursing, but microbiology is slowly changing my interest. I'm feeling my excitement rise when I look under the microscope and discover and learn more about microbes. Fascinating. I was wondering if we as students have access to the papers you talked about in the podcast. Particularly, I'm really interested in the one on biofilms. I saw the links at the bottom of the page. Are those the papers, or is there more? Well, Holly, you need to be curious and click the link and find out yourself. They're the links to the papers, but your library at your school may not have access. So,
but some of them will be at open access. So, one way to find out is to click.
And Holly, the paper that we just talked about for Parkinson's and is in the public domain, as is the original mouse study paper that we talked about as well. So you can certainly look at those.
The next one is from Ciro, who writes, thank you. The mentorship bias. Women are often backseated, and it's so wrong. Brings awareness. So that was a couple of episodes ago, right, Michele?
Yeah, where we talked about work done to identify what the expectations are at different types of academic institutions so that we can use it to better inform our searches for faculty, but also help trainees understand what kind of skills they want to acquire if they want to be competitive for a particular type of job.
And one from Jackie. I'm currently taking a microbiology class, and I listened to the podcast episode, Microbiology Papers for First-Year Students. The topic of phage therapy came up, and I was wondering, do you think phage therapy would be used more than antibiotics in the future? What problems do you see happening if it is commonly used? I think we got a course assignment here for the students to write into TWiM.
Cool.
Because this is the second one here. Michael, what do you think?
I'm going to give the answer. Stay tuned. We're going to have a paper on this topic in an upcoming episode. Phage therapy does offer some promise. I worked on it a number of years ago, and it was very successful at knocking out a Pseudomonas infection in an animal model. We'll see if we can get it to go. Now, with the advent of all this technology that has resulted from the development of SARS vaccines, I think we might be able to get phage therapy into a reality.
All right, that's TWiM 238, microbe.tv/TWiM for the show notes. If you want to send us a question or a comment, TWiM@microbe.tv. If you'd like to support us, if you like what we do, microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you, and take care, everyone. Got to hang on for just another couple months.
Elio Schaechter is at Small Things Considered. Thanks, Elio.
My pleasure.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Hope you stay safe through those
it just thundered.
Tornadoes.
I was looking out the window and going, is it getting really bad?
Well, we were able to get the recording in, so that's good. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.